Hi, this is Mr. Waite. Welcome to the third and final session, Saturday study session for biochemistry review. We're going to be looking at the free response questions this time. I am going to ask you, if you're doing this podcast through Play Post-it, I'm going to present to you the questions. You should also have the handouts in front of you. I would like you to type in a response when the video pauses and I ask for a response. It should be a free response area. Go ahead and type in something. Type in any ideas, type in something thoughtful. I would like to read through this and know that you put some thought towards it. You don't have to get it exactly 100% right. But again, show me that you are trying. And the other thing is, I don't know how other AP Bio teachers feel, but since this is just practice, I do feel that it's okay to outline your response. Just give a quick response, the topics that you would talk about. You don't have to go into great amount of detail. Just keep in mind you will never want to do that for the AP Biology test or the mock exam. You want to be as thorough as possible. Make sure you're writing complete sentences and not just an easy outline form. But just for purposes of practice, it is okay this time. So let's go ahead and get looking at these. Hopefully you have your review sheet with you. The last two videos we went over the multiple choice questions. We also did the two math grid-in questions. So now let's turn our attention to the first short free response question, and let's see what we can't do with that. It reads, proteins, or large complex molecules, are major building blocks of all organisms. Describe the chemical composition and the level of the structure in proteins. So guys, I teach my students to go ahead and circle the command word, which is describe. In other words, we can't simply just list or identify it. We have to describe what we're talking about here. And what are we describing? First thing is the chemical composition. Composition of what? Proteins. Okay. And the second thing we're looking at is the levels and structures of proteins. So there's kind of two things we're looking for here. We need to address both of those. So at this point in the podcast, what I'm going to do, actually let's go back and look at that one more time. I didn't mean to take that off the screen, sorry. Go ahead and if you're doing the podcast, pause the video or it will stop for you and do your best to answer these. And as soon as you get an answer in, we'll go back and see what the rubric looks like. So let's go ahead and do that. If I go down on the rubric, I'm going to have to go back over here, excuse me, and let's look at this. There was our question. It's a 5-point question. The first thing that it's looking at broke the question down into chemical composition. That's where 2 points came from. Guys, it was specifically asking about proteins, so anytime you're asking about proteins, think about the building blocks. Don't forget that amino acids are the building blocks of proteins. These are the monomers of the polymers over here. Now, if you're going to go into further detail, remember, write everything that you can think about. They went ahead and gave you points for describing the amino acids, the amino end, the carboxyl end, and how they each have their own R group associated with those as well. They went ahead and further said, what is the composition? They said amino acids are made of carbon, hydrogen, oxygen, and nitrogen. So any three of these, each bullet would be worth a point. There's a 2-point maximum here. The next part is the level of structure, and I know a lot of students sometimes have difficulties with these, so it's good to go back and review. Don't forget there's a primary structure where we're talking about just the basic amino acids being linked together by these peptide bonds. So here's one, here's one, here's one. Again, those bonds in between the amino acids are called peptide bonds. That's just like a straight line of amino acids. Next thing we do with that string of amino acids, we could fold it over. They often have a helix or a pleated sheet shape to them, and this is due to hydrogen bonds, hydrogen bonding between those. So I often think of it as here's a string, it's a string folded back on itself. Next one is the tertiary structure. And this one, that's the R groups on the amino acid that are going to be interacting. They listed different types of bonds here, hydrogen ionic bond, or even the van der Waals interactions. I like to think of this as taking my folded string like this and folding it over again. So it's starting to look like this. It's starting to get that three-dimensional protein shape. And then lastly, it's our quaternary structure. And this is important to recognize, 2 or more polypeptides interacting together. So it'd be kind of like looking at 2 of these things. So if this is one, here's another one that has that tertiary shape, and they're interacting to give it the quaternary structure. Here's a better picture for you. This again is a good image to keep in mind. Here's the pleated sheet image that they're talking about right here. But again, here's our amino acids connected by peptide bonds. The secondary structures, like wrapping that around your finger. Tertiary structures, like taking it off your finger and wrapping it back and forth a few times. And then finally, your quaternary structure is a few different tertiary structures interacting. Hopefully you were able to get some of those points, if not all of those points. Again, that's a 5-point maximum. Don't forget, FRQs are where you get the points for the AP exam. Not always easy, but don't be afraid to talk about all that you know about the particular subject they're asking you about. Let's go on to the next question. Let's go back to the sheet that you have. It looks like this. And this is talking about water. It says up here, water is important for all organisms. The functions of water are directly related to its physical properties. So again, I'm going to circle the command word. Let's get a color we can see. It says describe. So again, we can't just list it. We have to describe it. And what are we describing? We're describing the properties of water, how they contribute to 2 of the following. I like to put boxes around the numerical numbers to show me they've given me a choice of 3. I get to choose 2 of those that I could talk about. So it's either the role of water in metabolic processes, or it could be the ability of water to moderate temperature within living organisms, or how water moves from the roots to the leaves of a plant. Choose 2. Keep in mind when readers grade these, they will only grade the first 2, even if you talk about a third one. It's not worth your time to answer all 3. Answer 2 as completely as you can, and in the podcast I would pause the video now to give you time to do that. Let's go ahead and let's go back and now look at the rubric for this one to see what they were looking for. All right. This one was a 4-point question and describing water's role as a medium. To keep in mind, water is the universal solvent. It could dissolve a number of materials. And then you could have also talked about osmosis. And remember, osmosis is the movement of water, specifically across a membrane, because of the water potential differences. Other things you could have talked about: its ability to moderate temperature, keeping it in one spot, not getting too hot, too cold. You'd want to use the word specific heat or even specific heat capacity. Talking about in the environment, if you live next to an ocean, the water in the air keeps the cool during the hot days, keeps it warmer during the cool days, because the water is absorbing or releasing a lot of that energy. Could have talked about the heat of vaporization, how the cooling process, when water turns from a liquid to a gas, it needs energy to do that. Where does it get it from? Probably the organism that is sweating and giving that energy to the water droplet to be cooled off, thus taking away energy from the organism and cooling it down. You could have also talked about how ice forms along lakes or rivers, and that ice forming on top will keep the water below warmer in a liquid state, which allows aquatic organisms to survive. Last few things on here. If you talked about water moving in plants, the key word here is transpiration. You can't just list it, you've got to describe it. This talks about when the loss of water due to the stomata being opened, that's going to pull the water up through the tree and eventually out of the leaf, but it's creating a water potential difference. That's good to recognize. And hopefully you can see this one down here. It's talking about capillary action of water due to cohesion and adhesion. Remember, cohesion is water binding to itself. Adhesion is water binding to something else. So hopefully you were able to get some of those possible points. Remember, just getting half is a good achievement. But let's go ahead and let's go on to the next one here. And on yours, there's our formula sheet. Looks like we have one final long free response. It has two parts, part A and part B. All right, the effects of pH and temperature were studied for an enzyme-catalyzed reaction. The following results were obtained. They give you two graphs here. Make sure you always, anytime you're given a graph, look at your axes. The Y is enzyme activity versus temperature, versus this one is enzyme activity versus pH. You can see a typical bell curve, or type of a curve associated with these. The first one says, discuss the relationship between the structure and function of this enzyme and explain how the structure or function of enzymes are affected by temperature and pH. So again, I have my students go ahead and circle where the command word is to explain. Oops, I forgot one over here, is to discuss the relationship. And what are we discussing here? Between structure and function of this enzyme. So how does its structure affect its function? And then to explain how that structure and function are affected by the temperature and pH. That's part A. Make sure when you're answering this, you label it part A, and you answer the discuss the relationship, and then you explain the structure, how it's affected by temperature and pH, not just one or the other. And then we'll go on to letter B. Make sure you identify that to the readers that you're talking about B. And it says describe. What are we describing? A controlled experiment that could have produced the data shown for either or both of these graphs. So make sure you get a controlled experiment, guys. I tell my students whenever they see design an experiment question on FRQ, you should relax, you should be happy, because they're not necessarily easy, but they are easier. Just think about all the parts of the controlled experiment and make sure you mention those. It does, actually I was going to say start with a hypothesis, but it says down here to make sure you include a hypothesis. And don't forget, when you're doing that, we always write if, then, to identify the hypothesis. Keep in mind your different variables, your dependent and your independent variables over here. Make sure you identify those, and you have to make it specific to either pH or to temperature, how the enzyme activity is reacting to either of those. Other things important for controlled experiment designs, don't forget to make sure that you're doing this multiple times, that it's repeated so it's more valid. Try to think of everything you can. We'll pause, write a little bit, and let's go ahead and take a look at the rubric. All right, the rubric for this question, part A, what they're looking for here is an optimum temperature and a pH. In other words, identifying the top of that curve that we saw earlier. This is the optimum temperature. This would be the optimum pH over here. And keep in mind, enzymes, once they go beyond that, they tend to denature or change their shape, and they don't work as effectively. You could have also talked about the enzyme substrate complex, or how they fit together like a lock and key mechanism. This one says you could have talked about how the tertiary shape determines the function as well of the enzyme, and that's the shape that would be changing once it denatures beyond a specific pH or temperature. And a description of the enzyme structure or function, and they give you down here some of what those possible structures or functions could be. I'll let you look through those. They could have talked about this idea of, again, denaturing, and it said what would cause that denaturing, a change in the temperature or pH, and that would, what happens during the denaturing process, the enzyme activity decreases greatly, and again, that's because of that change in shape is for the denaturization. How temperature affects the conformation of things, what they're looking for there is keeping in mind higher temperatures tend to break bonds, specific bonds, so you would have to mention in this one the hydrogen bonds or the van der Waals force. How pH affects the conformation, as it says that, don't forget, what is pH measurement? pH is a measurement of the hydrogen ion concentration, and that specific that concentration is going to affect how bonds interact with one another, whether it be the hydrogen bonds or ion